Evo, dolazi ljudi, lijepo.
To mi je uvijek panika. Kad imamo predavača, ona hoće doći.
Ma razumijem. Pogotovo, ne znam, imaš iskustva ili je brojnije kad je subotom ili je recimo prek tjedna kada je recimo radni tjedan. Šta misliš kad ima više ljudi, nemam pojma, koje je tvoje iskustvo?
Nismo nikad proveli preko tjedna, jer mi nismo baš stizali preko tjedna. A ove subotama, ne razumijem. Kao, ne znam da li bi ja slušao da ne organiziram. Ali ono...
Jasno.
Pitanje kolege, da li će predavanje biti snimljeno da može posle ponovo da se pogleda ili kakva vam je ideja?
Predavanje će se snimati kolegica, druga koordinatorica će snimati, pa ćemo to sve na naš YouTube kanal.
Super, može. A da, zapravo to sam ja nešto se složio da se snimalo tako, već sam zaboravio. Tako da, bit će snimljeno. Super. Svakako, ako će iko imati pitanja, stojim na raspolaganju ako treba šta ponoviti. Mislim, nije ovo, kako bi rekao, atomska fizika. Malo smo se igrali sa protokolima, malo ih optimizirali, poboljšali i to je cijela priča.
Što se tiče predavanja, da li odabrati vikend ili radni dan, veruj mi, nema pravila. Za ono ko je zainteresovan, taj će naći vremena čak i u sred svog radnog vremena. A onaj ko nije zainteresovan, taj ti neće doći preko puta. To se meni dešavalo, kako se zove, znači dođu ljudi, prođu 50-100 kilometara da čuju neko predavanje, a koleginica koja živi preko puta zgrade gde sam bila ne dođe. Tako da nema pravila.
Da, razumijem, idete tako. Hvala. I da, lijep pozdrav, Gordan. Nismo se još, ne znam da smo se imali priliku poznati, ali evo da, drago. Drago mi je vidjeti arahnologe ovdje malo.
Znamo se preko radova, verovatno.
Tako je. Čuo sam o vama generalno i preko ove akcije, ovo što ste promovirali arahnologiju po Srbiji, to sve, svaka čast u vezi toga. To mi se jako svidlo, podupiram takve akcije.
Hvala kolega, trudim se.
Što se mene tiče, možemo polagano krenuti, ako si ti spreman. Odlično.
Može.
Super. So the lecture will be held in English, obviously, but I will firstly do a small, in my case, Croatian introduction as tradition by BioBalkan. Pa pozdrav svima, iznimna mi je čast vas pozdraviti na trećem predavanju treće sezone Bio Balkan Collab inicijative. Danas sam ovdje ja s vama, zajedno sa kolegicom Vanjom koja će snimati. Danas je s nama kolega dr. Domagoj Gajski sa svojim predavanjem o molekularnoj biologiji i paucima. So hello everybody, welcome to the third season and third lecture of that season of Bio Balkan Collab Initiative. Today with us is our lecturer Dr. Domagoj Gajski with his topic Facilitating Taxonomy and Phylogenetics, an informative and cost-effective protocol integrating long applicant PCRs and third generation sequencing. A long title but a very interesting topic that I can hear more about. If you have any questions during the lecture, feel free to write them down in the chat. We will read them afterwards, and obviously you can also ask through microphone after the lecture. I give the word to Domagoj.
Thank you. So let me just one more time open and share. And can you see my presentation?
Yep.
And can you see my mouse moving in case I want to show something?
Yes.
Perfect. Okay, so good afternoon, everyone. My name is Domagoj Gajski. Today, I will talk to you about how we, let's say in some way, improved the research of taxonomy and phylogenetics, comparing long applicant PCRs and third generation sequencing. This research was done under close collaboration of my university, where I did my PhD at Maastricht University, and at S3E3 University in Germany, where I worked under my then supervisors Henrik Winkel and Susan Kennedy. They have a lab there where they mainly focus on barcoding, but they as well develop a lot of other protocols. So I wanted to experience a little bit how is it to optimize and develop new protocols. A little bit about myself. I did my PhD at the Maastricht University, which you can see here, this logo here. And this mainly, most of this research here was done at the S3E3 University, but right now I work at the Vukas Institute, where I mainly focus on forest ecology. And it's a quite new field for me, but as well, hopefully in some future, I will have some interesting talks about it as well, what we are doing there. But about this today's topic, so what we are doing today, what we did actually, what we prepared and what we will talk about today is a protocol which we developed. But before we jump into it, I would like to talk about taxonomic knowledge. It's basically the essential element of any of biological fields, from ecology, taxonomy, from even molecular biology. It's good to know sometimes for what kind of organisms and whose even proteins and everything you work with there. But as well, it's usually this taxonomy faces challenges, as any field and any research usually. One is that there's usually a shortage of trained expert taxonomists. One other is that sometimes there's quite a shortage of fundings. And sometimes as well, the identification of characters which are needed to identify some organism to a species level is missing. Yeah, it's not available, especially, for instance, for juveniles of many arthropods and spiders, as you can see here in the picture. That's a really big issue here. And it can be even more challenging for taxonomy and phylogenies, which you develop when you have really super diverse taxa like spiders, which currently have, if I remember well, over 50,000 species worldwide. And regarding that, basically one method which helped as well in identification was DNA barcoding, which as well still is used to, in one way as well, identify the specimens. It's basically that you identify species and confirm their morphological identification based on DNA, based on usually some sequences. These barcodes are usually deposited in large databases through which you can then search your sequences to find to what they belong, for instance. And there as well, it's a method which is already quite standardized. It's getting quite cheap. It's usually $5 to $10 per sample. But one problem is that usually it's just one part of a gene, up to 600 base pairs, which has usually a limit of explanatory power. Sometimes it can't go to even species level. If the species are really closely related in their, especially these genes, which we usually use, like for instance for arthropods is the cytochrome oxidase 1 gene. So this can be then quite a trouble. And as well, we use sometimes, actually not sometimes, but I would say not anymore so much now, but generally before, DNA barcoding was used in phylogeny and is still now used in when the budget is scarce. It's basically that you use this sequences which you obtained, usually from 1, maybe 2 or 3 genes, to infer some phylogeny of closely related species in general to some extent. And it's as well, again, limited to one of several shorter fragments. Why is it a problem with shorter fragments? It's mainly because our sequencing technology, which is usually affordable, is limited by shorter ranges of sequencing. And then as well, because of the shortness, we can't explain the whole evolutionary history of a species and between species based on these genes. Sometimes these genes are usually not diverse enough to explain the evolution of them. So there are newer methods for obtaining information for phylogenetics. Some of them are here mentioned. These are basically the most used. One is DDR sequencing. It's basically that you using some specific enzymes lysate several special parts of the DNA, so that you get small fragments which you then sequence, and then you have fragments from all over the genome there, which you can then use for, you get a really huge amount of information, which you can use for each specimen to infer a phylogeny. Then you have UCE, which is ultra-conserved elements by targeted enrichment. It's basically that you look for regions which are really conserved, and then there you try to infer some phylogeny, especially for if you go really deep into the phylogenetic tree of big clades. Transcriptomics is another way where you basically extract the RNA and then try based on the RNA to get some information. And then we have as well the genome sequencing, which is basically you sequence the whole genome and then you compare genomes between each other. This is basically then you have the whole set of information and you can compare them. But as well, there's a problem with this. Actually, first, one good thing is that you have large amounts of data from all of these methods, from hundreds of loci. So you get huge amounts of information from different regions with different speeds of mutation. But the problem is that they're usually compared to barcoding, for instance, they are 10 to 100 times higher cost per sample. They usually as well require sometimes some modern, well-equipped molecular laboratories, and as well really high computational strength, which means as well costs on getting such strong computers or servers or anything where you can process all this data. They take time as well and money. This is why in the Trier lab, we decided to develop a new barcoding protocol, which can be as well used to infer phylogeny, where we basically have this methodology here, which you can see on the screen. I will go step by step through it, what we did actually here. So the first thing, what we did is, of course, you need some specimens to compare and to use for such a phylogeny. Here we use spiders mainly because I have broad experience with spiders and I can identify them as well to species. So I wanted to use them as a reference for this phylogeny, which we will infer. So we went to a site which is close by to Trier in a forest. You can see it here is Trier. It was literally just a site with a little bit of grass, bushes, and a little bit of forests, a little forest patches and stuff like that. So we realized there probably we would find the highest diversity of spiders in one little spot. So we went there just for one day, and we in one day managed to find 63 spider species belonging to 47 genera and 20 families. I will show you some of them later in the results. So we used then this spider species to test these PCR primers which we developed. As I said before, the previous barcoding primers and amplicons which you get when you perform a PCR were quite small, like less than 600 base pairs or around 600 base pairs, maybe up to 1,000 depending on what kind of Sanger sequencer you have for barcoding. These were usually used. Here we developed primers to amplify regions which are called cytochrome B, cytochrome oxidase 1, and the whole 18S to 28S gene region in the ribosomal section of the genes. And they are much larger. Okay, maybe cytochrome B not so much, but these here are much larger. They're in their size are much larger in size than a Sanger sequencer could ever sequence it. But I will tell you later why we decided to use those. So such big ones, primers, we had to download many as well genomes and sequences of spiders which we could obtain. We then developed many primers. You can here see all list of primers which we found around these sequences, which we could use there. There was a huge list of different, we tried even different regions and different sets of regions. We decided in the end to take those which were the best, like the CO1, cytochrome B, and 18S, 28S. These all primers were basically designed and then tested by me. So this was around probably 2-3 months of work to find all these regions and sequences on through databases. And then, yeah, of course, we were as well testing them on all these groups of spiders. So here is how basically we check if a primer is really valid. I downloaded a whole batch of all the available sequences of spiders of different groups. Here they are grouped into these bigger clades so that there's enough space here to show you. And then here in this little brackets, you can see the percentage of each of these nucleotides, which is available at exactly this spot in the gene. So you can see this primer, which I developed, matches this region really well. And there's basically no mutation here in the beginning, which is really a priority for a primer to work well. Well, the variety could be some variety later. And if you're confused, why is there a S or H or something? These are usually like a combination of different nucleotides in the same region. So, for instance, one primer has here just like, for instance, C, and another has just what was here? G. So, but they all have this beginning really similar. So this is what we actually designed. And then here are basically the regions which we chose. We chose the CO1 gene, which is around 1,100 base pairs here long. He is in the mitochondrial genome. You can see it here. And this is basically the size of this gene which we took and we were testing then is it's basically almost the whole gene. Yeah, it's a little bit like several hundred bases less than the whole gene. So we basically took the whole gene length to get as many formative sites as we could. Then we had the cytochrome B gene, which is around 750 base pairs, which is again as well almost the whole gene. And then we took this whole from the nuclear genome region of 18S to 28S, which is a really huge chunk of 4,300 base pairs. As you can see, it encompasses not only the 18S and the 28S, it has as well the ITS there, regions or the internal transcribed spacers, and the 5.8S ribosomes. So everything is in one place there. And here we have actually many regions of different mutation rate, so it could be as well useful for obtaining as much information as possible for phylogeny inference. After that, we, as I said, if you remember here, I can show again, we had hundreds of these primers, not hundreds, but basically hundreds of combination, but I already chose some better ones a priori by some logic, which we could test. And we went through probably, I don't know, somewhere around 500 PCR trials to test on all these specimens of spiders which we had, which are the best PCR primers to use. And then not only that, but we decided to combine them into one reaction to see which of these primers combinations work the best in one reaction, so that we don't need to waste a lot of money on many reactions to do for each of these genes separately. So we decided that we combine all these gene primers into one cocktail and then get the results in one cocktail immediately. And as you can see here, finally it worked after a lot of trials and temperature adjustments for PCR. You can see we obtained all three regions here from 4,300 to 1,100 and 750 base pairs. You can see this is basically, I don't know how much you have knowledge about molecular biology. You are free to ask if you have any questions. But you can see that the bigger the fragment, the higher up on the gel it is. This is a gel electrophoresis. And this way, basically, what we obtained is a total of around 6,150 base pairs of informative data, which can be used for a phylogeny. So we're not working on a small scale of 600 like with barcoding. Unfortunately, we are not working on a huge scale, but we are already on a scale which is 10 times bigger than the barcodes, regular barcodes with Sanger sequence. And so why did we develop such large primers which amplify such large regions? Well, because there's now this trend of sequencing everything with methods, with gadgets from the third generation of sequencing. If you are not familiar with third generation sequencing, it's that the first generation was basically the Sanger sequencing. The second generation are sequencing methods like Illumina, which is, I would say, the only one which still survived. And now we have nanopore, this here one, and there are as well several others which are in the third generation. What nanopore can do better than Illumina, he doesn't have a size limit. So you can sequence basically as long as you need to obtain the sequences of the length you want. I think the record was of several gigabyte bases, so it's several million, not even million actually, even more. Yeah, it's several billion bases. Yeah, so it's a really huge length. So this is why nanopore was really useful compared to Illumina or Sanger, which I mentioned here. And of course, you can as well run there many hundreds of samples at once, which is a huge advantage. So you save a lot of time and money when you can run everything in one gadget. And another thing is, as you can see here on the picture, it's a relatively small device, as you can see here as well. This is me working actually in the lab. I was preparing the nanopore sequencer. It basically looks like a little bit bigger than a USB stick, slightly bigger. So it's not really a huge gadget. This is the whole thing which you can see here. And what I'm pipetting here is the whole solution of, I would say, 150 samples, which we used with controls and everything. It's everything here in this pipette, and this tip here in the pipette. And a funny story was even here, I managed to do this in the office, like next to my computer. So I improvised the lab here. I put a little bit of ice. I had a shaker here and a little vortexer and some little bit spinner and some trash. Why? Because my supervisor at that time was sick. She had corona, because this was still around the corona time when we started this trial and everything. So she controlled me over the video camera. It was really fun experience. So yeah, this is what we did. Once you pour this in and you put some other solutions for better work of it and put some like controls into the solution, By the way, the protocol for nanopore sequencing is, I would say, quite straightforward. They explain you everything in instructions. So as long as you have prepared the sequences, it's really clear. And then you have this little device. This is how it looks like when it works. It has these lights which change a little bit all the time, like that it's really futuristically working. And here you can basically see, if you remember on the gel there, what I showed you, this amplicons of different sizes. You can even see them on the nanopore sequencer, how they are actually, these peaks are basically in their sizes. These sizes are slightly bigger than the amplicon sizes, just because the primers were added here. Yeah. So, and adapters were added here. Yeah. So this is the size of it. You can clearly see that the sequencing works, and then it sequences all the 3 spikes. So what is else there? Now we go to bioinformatical processing. For this, I think bioinformatical processing is for some really like abstract science, especially a lot of coding. This is why with my help of my colleagues, we adapted a script from a Nature paper for nanopore processing sequences. We adapted it for our protocol, so that once you get your sequences out of nanopore, you just you would need to open it in a Unix terminal. But if you have a Linux, then it can be done. You just then need to copy paste the code, add in the same folder where your script will be running, your folder with the raw sequences, and it will create you literally a little folder for each of your samples with the data inside. So you will immediately have everything there, all the sequences you need for processing later. Yeah, everything will be fresh and ready here. This is how it's creating here. And when we blasted our sequences, they were usually all the time a really huge hit, which tells us we are sure that they are showing the right thing. Because we had, for instance, this spider, and it showed that it was this spider. And then we tried to do a phylogenetic inference to see how it works. This was done by my colleague, who is a phylogenist. He aligned all the sequences of all the spider groups and spider species, and then he ran a maximum likelihood, which you can see here on the picture, and you have here as well the picture of all the spider species which we use. You can see here we had a really diverse all over the spider clade. We just didn't have tarantulas, for instance, in this big group. But all the tested, and then when we obtained this phylogeny, we compared it with basically, I think, 10 or 15 studies which used hundreds of loci with different methods, which we mentioned, like DDROT sequencing, UCE sequencing, genome sequencing. We compared them with them, and basically they were aligned with all of them on a level that they had all these big clades like Aranoidea, Maronoid clade, Dionycha clade, RTA clade. They all were monophyletic. The same was for the families like Pteridide, Linyphide, Tetragonatide. Again, monophyletic. So we were really aligned with all the studies. As well, the genus level was aligned, and even the species of the same, because we sometimes had specimens of the same species several times, they were always grouped together. Yeah, so it worked really well. And there were, of course, always some slight discrepancies, but they worked with one method of higher and slightly were a little bit different from the other. But the big picture was consistent with all of these methods, which basically says that you could use it for up to, I would say, family and maybe a little bit higher than family. This method could work very well, at least for spiders. For others, maybe the rate is for other clades, the arthropods and everything else can be a little bit different. And this is our barcoding protocol. As I mentioned, this protocol was designed for spiders, but can be, I would say, quite easily adapted for all the other arthropods. And if you want to even more, a little bit change the sequences, which you need some references, you can even use it for vertebrates later, yeah, because all these genes exist, so you could, in theory, use it. So it can be applied for basically any organisms, any animals, basically. And what is the advantages of this barcoding protocol? It's useful for taxon assignment, but as well for phylogenetic inference. It can be performed in an improvised laboratory. basically all this you could do even in the jungle if you have literally just an improvised PCR machine. Because my colleague actually tried to do it in the jungle, and it works. So, and then data output is not computational intensive. I think even the results you would get in several hours after you run them for the phylogeny, for instance. And definitely it's more user-friendly because we have now the instructions and the script which we wrote for running it. And what are the advantages? Our protocol costs $5 to $10 per sample. And gives you 6,000 base pairs of data per sample. Compared to Sanger, it's the same price, but gives you 10 times less data on average. Compared to Illumina, the same thing. It's even cheaper than Illumina. And compared to these big methods for a big amount of data, they are basically much lower, even 10 to 100 times less than the other methods. And although they don't give such a huge amount of information, they give a relatively reliable amount of information for recent for smaller level taxonomic level phylogenies up to family level, definitely. Yeah. So this is what we aimed to actually have affordable things so that phylogenists around the world could use it, who don't have the budget, taxonomists as well who could use it, who don't have a budget for it. And I hope in the future it will be used more. I've seen already several people contacted me about some ideas how to use it, and I give them advice. So hopefully in future there will be more studies. And this way, I would like to thank you for your attention. I would like to as well, one more time, thank my colleagues who all a little bit of their knowledge incorporated into this development. Here is a QR code of the paper if you're interested in reading it. It's published last year. And yeah, thank you for your attention. If you have any questions, I'm ready to hear. Okay.
Thank you for your lecture. We will read the questions now. I hope there are a few. So there's a question in the chat. Very interesting protocol. Could this protocol be adapted for prokaryotes?
Prokaryotes? Yeah, basically, I mean, in theory, maybe let me show this slide so that it's better. In theory, you would, for prokaryotes, need to adapt definitely the primers, but you could basically use. So I don't know which are the main some common genes which are used for interfering the phylogeny of prokaryotes. You would need to find there and then just align it, but. You would need just some reference of many results where, for instance, you have really long sequences so that you can find regions for the primers to attach. And then, if you want to design primers which are really long for several thousands of base pairs, you could do it, without a problem, It can be done. If you need more help with it, we could have a meeting and we could discuss it. I would just need to have more knowledge about what are some common barcoding regions there, and what is everything sequence. So what we can use as a reference for building some primers which will amplify long regions for such phylogeny.
Thank you. And there's also the supervisors. Ever learned to stop micromanaging their cohort remotely? I suppose that's a joke. Another question is, thanks for a nice talk. I had two questions. Would it be possible to amplify such long regions, 4,300 base pairs from older samples? And what is the error rate for sequencing such long fragments using nanopore?
Would it be possible for old samples? Hardly, hardly. This is probably something I forgot to say. Definitely, this is not a method you could use for museum specimens. Just forget about it. But for any kind of fresher material you have, so we worked with even samples which were up to 5 years preserved in ethanol and everything. So It can work, yeah. It can work. This 4,300 base pair maybe will not immediately. I think I see another question. I already answered it. So I would say up to 5 years preserved in ethanol, and that was freshly preserved in ethanol. Like you catched it and then you placed it into ethanol. It can work. Yeah, it can work. We had in some cases that this long primer for some spider specimens older didn't work. But I would say it's we tried it. Funnily enough, we had for one spider of the same species it didn't work for, but for the other spider of the same species it worked. So it probably depends a little bit on as well how the material was stored and everything, because one was actually a little bit older. But generally, the smaller ones, let's say up to 1,000, 2,000 base pairs, such primers could work without any problem even for this. And you can then just develop it on some other gene regions, which you as well found in studies. So I think for arthropods, from my experience, arthropods, there are at least 7-8 such barcoding regions which can be used for such things. So it could work, yeah. It can work, yeah. For these older ones, but for fresh, it definitely works. 4,300 base pairs works, yeah. And then, what is the error rate for sequencing for such long fragments using nanopore? So yeah, everybody was afraid about this error rate using nanopore. But the thing is, first of all, the newer gadgets which they release every year, their error rate is now 99.9%. So I did this on a 99% error rate, this one. And what how you fix the error rate is that you need to make, when you sequence it, so depending on the number of samples, you will tell the nanopore sequencer that you want a sequencing depth of, let's say, 5,000 reads of sequences per sample. When you have, I actually, from my experience, what we tried, even 1,000 reads is enough per sample. The more, the merrier, the better. So what you get with such depth of reads per sample is that you can then align them all together. This is basically a script does it for you. And then he reproduces the final sequence for this gene, which was based on 5,000 sequences, which each has some errors somewhere, But then, because the errors are random, so then most extent they are random. Sometimes maybe on the beginning and the end there are some errors bigger, but through the bigger chunk of the sequence, then the errors are corrected based on a huge depth of sequencing. So this is how you can correct it. So yeah, I hope. So then you had a follow-up. So you use some other samples as well, not just the ones. So for this publication, we used only the fresh ones, yes, but we as well tested it a little bit on older ones. We wanted to see a little bit, and we wanted to see how well it could work, actually. Yeah, we wanted to see. But to be consistent for this publication, we used fresh specimens. Yeah, we used fresh specimens. Because, we wanted to be consistent. So the older ones were used for testing to what depth, to what time frame it could be done. Yeah. But to my experience, can go to up to 5 years. Yeah. So okay, I think I answered all the questions in the chat, right?
There's the question with the formalin. Would the spiders from the traps with formalin, would that be sustainable for the method?
No. From my experience, no. Formalin is definitely not working with it. Generally, formalin specimens are not especially for such long fragments, you wouldn't manage to obtain it. Maybe short ones, but like up to 1,000, but no. From my experience, if it's preserved in formalin, it wouldn't work. So it's suitable only for fresh or specimens up to 5 years, but which were preserved in ethanol. Yeah. Now, of course, it helps if they were even preserved and they were stored in a fridge. This even better helps it. But we tested in the samples which were really old and were not in rich, but were in atoms. So that's the important thing. They were in 70%.
And there's similar questions. Would samples from pitfall traps work? And follow-up question on that: would pitfall traps with stuff like propylene glycol work?
Propylene glycol. Well, depends how long it was in the propylene glycol. Yeah, so it really depends. We actually not for this, but I had a previous experience with something else. I was amplifying for my colleague as an additional lab work. Depends, I would say, literally how long they were there. Yeah, depends how long did you leave the traps outside as well. Yeah, this has a huge effect. Or depends actually at which period. Like if it fell, I don't know. If it fell there and it was in the propylene glycol just few days, I think it would be manageable. Yeah, but more than that, I don't think so. Yeah, the DNA degrades really fast, especially in spiders. Maybe in some more with a harder exoskeleton like beetles or something like that could be better, but with spiders definitely not. Yeah, spiders are basically just a liquid bag, how do I say, which is degrading really fast. Yeah, really fast. So I hope I managed to explain. So where can nanopore sequencing be done at the moment? Well, nanopore sequencing can be done basically everywhere. The thing is you need to buy one. And now if you're curious, what's the price? The price is basically the device, this one which I showed you, costs around, I would say, $1,000 or $1,500. So it's not actually quite expensive compared to a whole Illumina device. It's like a little small cost. Yeah. So it's just imagine it like a really expensive USB you would buy. Yeah. So it costs around $1,500, and then around $1,000 costs these platforms for sequencing, which you should, in theory, change every 2-3 times after using. So they are usable for 3 big sequencings. So let's say you have 3 sequences in each 150 samples, and then you would need to throw it away and buy a new this platform where you pour the liquid on, yeah, for sequencing. So I can maybe show the picture. I will show you just a second. Let me go there. So you see basically this black part here, which is here attached by this gray part here, is basically what you would need to change every, let's say, 3 to 4, maybe 5 times. I used it once 5 times, but the 5th time the results were a little bit already not the best. Yeah. So these are basically here, these little pores. Nanopore is called nanopore because it's developed little protein pores, artificial proteins with pores through which the DNA in its whole length goes through it. And then based on which nucleic acid goes through it, it develops a little bit of a different electrical current. And this is what the little microchips detect there. Which is amazing that we have such technology on such a micro level. So, but yeah, it can detect the change in the nucleotides, which go through the pore, and then it can detect the change. But these pores get clogged really quickly. Like after 3-4 times of using, they get more and more blocked. And you can actually check it on because when you buy a nanopore, you get their app. Where you can then, check how clogged your platform is. So it will tell you, like, you still have, let's say, 50% of your pores usable. I wouldn't use it if it's less than 50% because then usually it's not usable anymore. Yeah. So just a sec. So where did I stop with the questions?
So the next one is, for the first sequencing of a species, the first COI barcode, do you think nanopore is accurate enough?
Yeah, definitely. 100% it's accurate enough. If you make, as I said, if you make a sequencing depth of 5,000 reads per sample, which you can arrange by just leaving the how you actually arrange this. You just leave the nanopore running, and you just leave it running. And 5,000 reads per sample you could obtain in, 15 to 20 hours, something like that. So yeah, you can obtain it, yeah. And then when you have such a number of reads of the same sequence like CO1, same gene like CO1 of 5,000 replicates, which each has some small errors, but when you combine them all together and then take the average, it's accurate enough, definitely. Accurate. I've seen already studies using it as barcodes. For barcoding, I've seen it for many things. Even now, my colleague and I are chatting how to improve it for even meta-barcoding, of environmental samples from gut content to leaf content and stuff like that. Yeah. So it's possible. Yeah, definitely. And then there is a question from you, Davor. How do you expect that the data acquiring processing will look like in the future? Will it be even more simplified, accessible? Data acquiring. You mean like a general term? You mean the data?
Yeah. Those are questions from the side, from other colleagues. I believe the colleague meant general-like sequencing. So nanopore is the newest thing. How do you think the technology will develop further? Will it be even simpler, more accessible, and in that manner?
We all here, especially if we listen to some molecular biology and sequencing and bioinformatics lesson, I don't know if you ever listen to those, that the sequencing cost is drastically going lower and lower and lower, but I think it went to threshold. Yeah, it stopped. And now I think it will just increase because of services. So of customer service and the app development and everything. I think we got to some kind of threshold for now. Maybe there will be in the future some new development. But generally, accessible, I would say that even if a device is €1,500 costs, it's extremely cheap. I would say this is the most accessible we will ever get for now. Maybe I wish I am wrong, but it's quite accessible. So I would say that this is definitely, regarding this, it's definitely the most accessible now. Simplified. Well, I would say it's quite simplified already. Of course, you need a little bit of molecular biology background to prepare the PCR, to extract the DNA, although there are all protocols now for everything. So if you just strictly follow them, I think you can do everything. Yeah, we even explain the whole protocol in our paper. So if somebody wants to go through it, they can find it there. Yeah. And will it be more simplified? I don't know. I'm not sure about this one. Yeah, I really don't know about this. And then, have you had cases where DNA information showed different taxonomic plays than previously thought?
I believe the call. Yeah. Okay.
Yes. No, actually, we had some small discrepancies with some of the studies which worked on higher amounts of loci, which sequenced a lot. But it was mainly on the species level, not on the higher levels, like these genera family and these little clades, which some families form. It was mainly, and this actually happens not, I would explain it more because if you want to have a proper phylogeny on a general level, you should have as many species as possible, basically, of this genera altogether, so that you can make these little detailed branches as good as possible. Yeah. So we had some small discrepancies here, but when we tested it on these bigger ones, everything was matching. Yeah. So.
Thank you. Obviously, it's a popular topic. There's a lot of questions. Is there any other question? Feel free to write it down or even ask through microphone. Okay, so Gordon asked DNA primers cost. Tell a little more.
Okay, let me remember it. I don't know, never really all this by heart, but so DNA primers, especially for nanopore, would cost let me calculate it. Actually, I will take my calculator quickly. Just give me a sec. Because we usually calculate it by, let's say I have 0.2 cents per base, something like that. And the primer would be around 40 base pairs. So it would be €8 per primer, maybe a little bit more with delivery. So each primer, and you would have enough primer there to run, I guess, definitely more than around several hundred specimens. What primers are, they basically are just, if that's as well what interests you, is basically they are needed to amplify a specific region of the genome of our interest. I made my primers. I can show you again. Primers can be really specific for one gene, and then they're usually just specific for one species. But here, the primers which I developed they're called degenerate primers or universal primers. You can call them as well universal primers. Why? Because they usually amplify a really broad range of related organisms, like here for spiders, for instance. Yeah. So they amplify really well the whole range. And why they are degenerate? Because I changed here instead of that it's just a clear nucleotide of these four main ones. I designed it like that there could be a switch here, that some of these little primers in the solution will have, for instance, here A in R, and some will have G. Yeah. So it will be mixed between A and G in this region here, so that they are more. I have a higher chance of obtaining this region for this specimen and everything. Yeah. So this is why it's designed like that. If somebody ever needs any help with primer design, luckily for this I have already huge experience. I do it by hand or by head, because I haven't found yet a software which could really reliably make me such degenerate primers really well. So which could be used for a really high, a diverse group of organisms. Yeah. So that's why. Yeah. But I of course I'm open to collaborate or help. You can always ask. Okay.
Thank you so much. Is there any more questions for the lecturer? Going once, twice, and third time. We will leave if it's fine with you, we can leave your contact on later. We can send it to the people, so you can ask him whatever questions you have. Thank you so much for the lecture. If there isn't any more questions, I would like to close this lecture. Thank you so much, everybody, for coming. Thank you, Domagoj, for this amazing lecture. And
thank you for having me, and for all the questions. I'm really flattered. Thank you.
It was a pleasure. Feel free to follow us all on social media for our future lectures. Thank you.